So today we'll discuss in detail about the connective tissue. Now if anyone asks you what is this connective tissue particular, what is the meaning? So from the word itself you can say what is the meaning of this connective tissue. Okay. So by definition the connective tissue is the tissue that connects, separates and supports all other types of tissues in the body and fills the interstices between more specialized elements. Okay, which serves to hold them together and support them. That means it is a type of tissue. What it will do? It will bind, it will separate, it will support. Connective tissue, it connects, separates, supports all other types of tissue in our body. Along with that, it fills the interstices between the more specialized elements which serves to hold them together and to support them. Okay. So not only it is binding, separating, but also it is filling the interstices. Along with that, it is also providing support to the different tissues, along with the different organs in our body. Okay, so that is the connective tissue. Now, like any other tissue, it consists of cells surrounded by a compartment of fluid which is called as extracellular matrix. Now, here the fluid means not the water. Okay, here fluid means it is fluid but it is having gel-like consistency. It is gel-like. Okay, so what happens in case of connective tissue? It is having a extracellular matrix which is situated outside the cell. Extracellular means outside the cell, intracellular means inside the cell. Okay, so it is having extracellular matrix or also called as ground substance which is having gel-like consistency, and this particular will surround the different types of cells which are present around it. Okay. Next, the connective tissue differs from other types of tissues that the cells are loosely arranged. Okay. Now, what is the meaning of this? I hope you have seen in case of epithelial tissue how the cells are oriented. For example, if you take the example of simple cuboidal epithelium, simple columnar epithelium. Come first. So in case of this epithelial tissue, what you have observed that the cells are arranged, okay, in such a way that the intercellular space is not there. Right, I hope you have observed that. If you take the example of simple columnar, simple cuboidal, simple squamous epithelium, in between the cells, the gap is not there, right? And those cells are attached to basement membrane. But here, cells are present, but in between the cells, huge gap is there. Okay, that means cells are dispersed in a medium of fluid, which is having gel-like consistency. Okay, and along with that another important element is present, and that is the fibers. Okay, different types of fibers. Next, if you see the structure of connective tissue, okay, if you see the structure of connective tissue, so in a structure of connective tissue mainly it is made up of three components. Entire connective tissue is made up of three components. What are they? As you can see, cells, fibers and ground substance. Cells, fibers and ground substance. These are the three components of connective tissue. That means any connective tissue in our body, okay, it will be having these three components. Now, depending on the area, okay, depending on the area, there are some variations. But basically, these three things are present, okay, which is constituted in the connective tissue. Now, if you see many tissues and organs in the body are made up of mainly aggregations of closed packed cells, for example, epithelia, solid organs, liver, etc. In contrast to that, the connective tissue are widely separated and a prominent intercellular substance is there. Okay. Next, this substance is in the form of ground substance within which these fibers and the cells are present. Okay, so that is the orientation of the connective tissue, how the connective tissue is present in our body. Okay, and as I said, these are the components. Okay, now I will show you a diagram, okay, a schematic diagram of connective tissue, so that whatever we have discussed, you will understand by looking at the diagram. So I hope you can see the diagram, everyone. Yes, so in this diagram, what is your observation? What you can see? Have you seen this diagram before?
Yes.
Yes, very good. So in this diagram you can see the ground substance. Can you see the ground substance? Yes. After that, can you see these are the cells? You can see these are the different types of cells are present. Now just compare between the difference. Okay, compare the differences between epithelial tissue and connective tissue. Okay, so already you have studied the epithelial tissue. Just see the difference. What is the difference in the orientation? Okay, so here in between the cells you can see spaces are there, right? Next, can you see these structures? This yellow color structure, this black color structure. Actually, such colors are not there. To make you understand, these color codes are given. Can you see these things? So what are these? Fibers. Okay, these are fibers. Okay, so see here you can see one type of fiber, here you can see one type of fiber, here also you can see one type of fibers. Okay, so different types of fibers are there. Now these things are constituted in a gel-like substance that is called as ground substance. So this is the ground substance. Okay. Now generally what happens whenever you are preparing slide in order to see the ground substance, okay, so the constituency of the ground substance such a way that it is to wash out. Okay. In preparation of the slide there are different steps. So whenever we are preparing it, generally it is to wash out. That's why it is very difficult to stain that particular part. And unless and until you stain anything, it's not possible to see under microscope, normal compound microscope. In order to see anything under compound microscope, you have to stain it. Okay. So that's why it's very difficult to see the ground substance. So why I am telling you this? Because in the practical class, okay, in the practical class when you will see the slides of the connective tissue under microscope, at that time you won't be able to see this ground substance. Okay, because here we are not using electron microscope and all. Here we are using the normal light compound microscope. Okay, this type of microscopes are used. So in this type of microscope, and I am having one slide, I will show you the example of one connective tissue. Okay, so here it's very difficult to see the ground substance. As I said it is washed away. If it is washed away, it will not be able to take the stain. If staining is not there, you can't see anything. Okay, but you have to assume that yes, this ground substance is present, and this ground substance is holding the cells and fibers in its position. Okay, now if I go back to the components of the connective tissue, as I said, three components are there: cells, fibers and ground substance, right? Now in order to understand in detail about the structure of the connective tissue, first we have to see about the cells, we have to study about the cells, then about the fibers, and then about the ground substance. Okay, so what do you think, what will be the different types of cells present in the connective tissue? Any idea? Yes, very good. Mast cell, very good. Macrophages, very good. Okay, so many different types of cells are there. Now what we'll do, we'll categorize them. Okay, mainly two types of cells are there. One is the fixed cell, another is free cell. Okay, or also called as wandering cell. So fixed cell means they are fixed, permanent. Okay, that means permanently they are present in the connective tissue. Free cells means they are not the part and parcel of the connective tissue. Okay, they are coming from blood stream and then they are going into the connective tissue. Okay, so these are the free cells. So here you can see the list of the cells. Okay, so as I said, two types of cells are there in the connective tissue. What are these? Fixed cells and free cells. Okay, now in some books it is written as free cells, in some books it is written as wandering cell, WANDERING. Okay, so don't get confused, दोनों का मीनिंग सेम है. तो if you see the list of the fixed cell, first you can see fibroblast, oblique fibrocytes. Now don't think that fibroblast and fibrocytes are same. It's not same, though it's written like this, but doesn't mean that both the cells are same. We'll see what are the differences. Okay, so if anyone asks you whether fibroblasts and fibrocytes are same or not, your answer will be no, these are not same. Some differences are there. I will show you the diagram of fibroblast and I will show you the diagram of fibrocyte. By looking at the diagram of the cell, you can differentiate. Okay, both the cells are not same. Next, you can see macrophages, also called as histocytes, then mast cell, adipocytes, also called as fat cells. Okay. These adipocytes, while doing the dissection, have you seen yellow colored things after removing the skin and all? Everyone, you have noticed that? Okay, that is nothing but the adipose tissue. That is nothing but adipose tissue. And it is made up of these adipocytes, fat cells. Okay. Next, pigment cells are there, also called as chromatophores, and then reticular cells. So these are the list of fixed cells. Next, here you can see these are the list of free cells. Okay. So free cells, if you see the list, I hope you have studied all those things in physiology. Right? Just see the list: neutrophils, eosinophils, basophils, monocytes, BNT lymphocytes, and plasma cells. So these are nothing but the cells of WBC, which already you have studied in physiology. Okay, so they are present in the blood, you know that, right? To provide immunological support in the various part of the connective tissue, from bloodstream they are coming into the connective tissue. That's why they are called as free cells or wandering cells. Okay, so we'll be discussing more about these fixed cells, right? Now please have a look. This is a beautiful diagram where you can see different types of cells present in the connective tissue. This is how they appear. Okay, but for this, if you use normal compound microscope, it is very difficult to see this type of picture. For that, we need higher magnification, higher resolution in order to produce these images. Okay, so you can see fibroblast. Can you see the fibroblast? Yes. Can you see the fibrocyte? So have you seen the difference? So this fibroblast, these are mature cells, active cells. Now these fibrocytes, these are immature or inactive cells. So first fibrocytes will be there, and after that it will become mature, the size will increase, and then we will be getting the fibroblast. Yes? Okay. Okay. Next, you can see the plasma cell. Okay. So why I am showing you these cells? Why I am showing you these cells? Yes. Yes. Another important point is there. As I said, when we will see the slides of connective tissue. Okay. Now this you have to understand. When we will see the connective tissue directly, you will not get any slide slide of connective tissue. Okay, if you go to the histology lab, one box is there. Inside that slide, permanent slides are kept. If you open the box, you will not get any slide directly written as connective tissue. Okay, connective tissue you have to study in the form of example. Okay, directly you won't get, because I am having one slide over here. I will show you. So here the slide is of adipose tissue, where adipocyte cells are there. So this is an example of connective tissue. Next, I am having some slides of ligament. I am having slides of tendon. Okay, so there also connective tissue is there. Now that connective tissue is there where you can see the collagen fibers. Okay, so directly connective tissue slide is very difficult to get. So you have to study connective tissue with the help of those examples. Right? Now, next important point why I am showing you these things are because directly you won't be able to see these cells in those slides. Okay? Directly you won't be able to see those cells in that particular slide. So that's why also I am showing you all those things. So now, though you have seen these cells, then you can see eosinophil, neutrophil. This already you have studied. Then you can see lymphocytes. These are the pigment cells. Okay? Then these are the mast cells. Next, before going to the next part where we will be discussing about the fibroblast, fibrocyte, all those cells, okay, under microscope, whenever you see any structure, which color is visible? Purple and pink. Okay, purple and pink. Now, do you know why? Yes, HN stain. So, in general, okay, mostly, if you see, mostly slides are made up of H&E stain. Why? Because it is easy to identify. It is easy to identify, but it doesn't mean that all the cells, all the tissue will take that stain. There are some cells, there are some tissues which don't take all those stain. Okay, that means if you put H&E stain, they will not take the stain. You can't see anything. For the different types of staining methods are there. Different stainings are used in order to stain that particular tissue or a particular cell. Okay, best example is this one. Reticular Marsh cell is there. Then reticular cell, I think it's not there. Okay, so those cells will not take H&E stain. For that, what we have to do, we have to use a different type of stain, and those stain are made up of silver nitrate, okay, or silver salts. So that's why the appearance will be different. And I am having some diagrams I will show you. Okay, so H&E diagram already you have seen. You know what is the appearance. Next I will show you the stain by silver salts and what is the appearance. So which is the first cell we have to discuss, that is the fibroblast and fibrocyte. Okay, so fibroblasts are the most common type of cells present in the connective tissue. Most common type of cell means if you take any connective tissue, fibroblast is the most common type of cell present over there. Now, what is the function? Why it is present? So mostly these cells they produce a diverse group of products including the collagen fibers, then proteoglycans, fibronectin, laminin, glycosaminoglycans, metalloproteinases and prostaglandins. Okay, these are the products, or you can say these are the substances which are produced by this particular fibroblast. Now, in the beginning, I hope you remember that I have told you that three components are there in the connective tissue, right? Cells, fibers and ground substance. So that ground substance which is there in the connective tissue, that particular ground substance is also produced by this particular fibroblast cells. Okay. Next, these fibroblasts are generally remain quotient, or you can say quiet, unless and until they receive some stimulus. Okay, then only they will start synthesis and they will start production of all the substances whichever I have mentioned over there. Now, fibroblasts are mesenchymal in origin. So the fibroblast cells which are present in our body, they are mesenchymal in origin. Okay, that means mesodermal origin. Cells part, and after that you can go. Okay, please don't mind. So fibroblasts, as I said, these are mesenchymal in origin. That means they are mesodermal in origin. If you see the shape of the fibroblast cells, as I have shown you earlier, they are elongated spindle shaped or stellate shaped. Along with that, multiple cytoplasmic projections are there. Okay, this is the particular shape of the fibroblast cell. Okay, next within the cytoplasm, if you see the cytoplasm of the cell, then rough ER is there, abundance rough ER is there, along with that large Golgi apparatus. So these are some important striking features of this particular type of cell. Next, they also produce reticular and elastic fibers. Okay, so along with the collagen fibers, they also produce reticular and elastic fibers. When they are associated with the reticular fibers, they are also called as reticular cells. Okay. Next, fibrocytes. So I have told you that fibroblasts and fibrocytes are not same. Some differences are there. Okay, now what is this different? Now, fibrocytes are inactive form of fibroblast. Okay, that means they are immature. They are smaller, thinner and morphologically different from the fibroblast cell. Next, macrophages. Already you know what are macrophages, you have studied. These are called as histocytes. Now the main function is phagocytosis. Okay, already you have studied. Next, mast cells are there. So they are small, round or oval shaped, and they are also called as histaminocytes. I hope everyone is aware of histamines, right? Which are precursor of allergic reaction in our body. Okay, so all the anti-allergic drugs or pills, whatever you are taking, they are antihistaminics. Okay, that means they will inhibit the secretion of histamine. Mostly they target the mast cell. Can anyone tell me another cell from which the histamine secretes? Physiology? Basophil. Okay. Next, okay, now after mast cell, next is the fat cells. So fat cells are also called as adipocytes or lipocytes. Okay, if you see the fat cells, they are, if you see the shape of the fat cell, they are signet ring appearance, the ring which we wear in the finger. Okay, everyone you know. So the appearance is like this. Now if anyone asks you why the appearance is like this,
because of the fat molecule.
yes, very good. Because of the fat droplet which is present inside the cell, it will push the cellular organelles towards the periphery. Okay. Next, whenever we are preparing the slide, during the stage of xylene wash, it is to wash away. After that, whenever we are staining, nothing is there inside the cell. So it won't get stained. But the cell membrane and the nucleus will take the stain. That's why it appears like this. Okay. Here I have shown you the diagram. Just see this one. Can you see signal ring appearance? Okay. So if you see in the center, nothing is there, because fat droplet was present and it washed away. If nothing is there, no stain, it will not take the stain. So stain is taken by the cell membrane and the other cellular organelles. Okay. And lastly, the pigment cells. So these are stellate shaped and can be easily distinguished as they are having brown pigment that is called as melanin. Already you know that in their cytoplasm. Most abundantly they are present in the connective tissue of the skin. Okay, connective tissue of the skin, choroid, iris and eyeball. Okay. Now, anyone, do you know what is the function of this melanin? Yes, very good. Along with that, it is also having another important function.
Protection against
Yes, protection against UV. Yeah, very good, very good. So, see one thing nowadays. I hope you have seen mostly people using sunscreen lotion.
Yes.
Okay. तो उसका फंक्शन क्या है? It inhibits the production or spread of the melanin, so that the pigmentation will not be there. Okay, which is bad. If melanin production is there, actually it will bind with the tyrosine. Okay, and then the melanin will spread in that particular area. So if you go to sun, okay, you'll observe that the particular area is becoming dark. Okay, because of the spread of that melanin. Okay, in that tyrosine is also helping. So as a result, what happens? The UV, harmful UV rays will not be able to penetrate the skin and go to the deeper structure. Once you are using the sunscreen lotion, what it does? It inhibits the process, natural process. Okay, as a result, pigmentation will not be there, but harmful UV rays can penetrate deeper into the tissue, which is harmful. Okay, so the best way is, instead of wearing all those things, putting all those things, just cover it, so that the rays will not hit the skin directly. That is the best way. Okay, so these are all about the cells. Next, fibers. We will not go into the fiber, just I will show you the fibers. So how many types of fibers are there in our body? 3 types of fibers are there, already you know, collagen fibers, elastic fibers and reticular fibers, right? Now, what do you know about the collagen fibers, anyone? Wait. Collagen fiber, what do you know about the collagen fibers? No, elasticity is provided by elastic fibers. Tightly?
Keeps the cell in position.
Okay. Now, in order to know about the each and every type of fibers, first of all, you have to know the individual fibers. Okay. Now, in case of collagen fibers, always they are present in the form of bundle. Collagen fibers, always they are present in the form of bundle. Inside that bundle, if you see the fibers, okay, they will run parallel to each other. Individual fibers, okay, they will not branch or anastomose. Next, if you see the elastic fibers, they are single fibers. Now these fibers can branch anastomose with adjacent fibers. Okay. Next, if you see the reticular fibers, here also they are single, but they branch anastomose in such a way that they will form a network-like structure. And these reticular fibers are nothing but the type 3 collagen fibers. Now the question is, how many types of collagen fibers are there in our body? Yes? Any idea? How many types of collagen fibers are there in our body? Approximately 20 types of collagen fibers are there in our body. Okay, among them, first 5 collagen fibers are there in your book. I have seen. If you take any histology book, 1 to 5, it is there. Okay. Now, other collagen fibers are also there, but those things are not given. Why? Because still researches are going on. Okay. So, among them, the third type of collagen fiber is nothing but the reticular fibers. Now, just see the variation. As I said, in case of collagen fibers, you won't get any branching pattern. Bundles can branch, but single fibers will not branch. Okay, they will run parallel to each other. But in case of third, the variation you can see, they're in the form of network. Okay, they're in the form of network. And these reticular fibers are forming the framework. They are forming the framework of most of the lymphatic organs of our body. Okay, for example, lymph node is there, spleen is there. Okay, so the framework of those particular organs are formed by the reticular fibers. Okay. So in reticular fiber and collagen fiber, elastic fibers also, so many things are there in detail. Okay. Where I'll be using some EM structure, or also called as ultrastructure. What is the meaning of ultrastructure? Anyone? Have you heard ultrastructure? You have heard about microscopic structure, right? What is the meaning of ultrastructure? Ultrastructure, the meaning of the ultrastructure is electron microscope structure, or the image of electron microscope. Now here or previous one, whatever I have shown you, these are the normal compound microscope images. Okay. Now let me show you one electron microscope image. Can you see the image? This is the image of which fibers? Collagen fibers. This type of image you won't be able to get in this particular microscope. Okay. This is of This is of electron microscope. Just see the fibers, the appearance. Okay. So as I said, always they are in the form of bundles, you can see. And can you see the individual fibers? Okay. So this is how the collagen fibers appears. Now you have seen this image is of electron microscope, and they are also called as ultrastructure. Okay. Now I will show you a compound microscope image. Can you see the fibers? Okay. So this particular type of image you will see under this type of microscope, light compound microscope. Okay. Now here also I have used one tissue. Which tissue is this? Ligament. You have studied about synovial joint. Okay. Synovial joint, I hope you have studied, yes. In synovial joint you have studied about the structure of synovial joint also. So in structure of synovial joint, you have seen ligaments are there, two types of ligaments, true ligament, false ligament, right? So if you take the TS of the ligament, if you take the TS and if you take the LS of the ligament, this is the slide which will be produced. And here you can see these are the bundles of collagen fibers. Okay. So under microscope, the previous one you won't be able to see over here. You will see this thing. And in the exam, by looking at this, you have to identify. So please keep that in mind. Repeatedly I am telling you, for connective tissue you won't get directly the slide of connective tissue. You will be getting examples of the connective tissue. Okay, examples. From there you have to identify. So this is one example. Next you can see the fibroblast also. Can you see the cells? Okay, so like this you have to identify. So these things will get clear as I will discuss everything about the connective tissue, because till now I have not finished the structure. Once the structure is done, then we have to discuss about the classification of connective tissue. Okay. 3 types are there, but we'll take 2 types because the mucoid or mesenchymal one is present in the embryo. Okay, so we'll skip that. Mainly we'll be taking the 2 classification, that is general connective tissue and specialized connective tissue. Okay, so when we'll discuss all those things, at that time also I'll show you some this kind of slides, then things will get clear. And moreover, you have to practice. Okay, you have to practice in the histolab. You have to see the diagram, you have to practice. Okay, these things are very important because during the time of exam, spottings are there. I hope you know that. Right? Slides, microscope will be kept like this, approximately 10 microscopes. In 10 microscope, 10 different types of slides will be there. Okay, and if you see, this type of slides will be there and it will be covered. Something is written over here, right? It will be covered like this by white paper. You can't see. Okay, and you will be getting, I think, 1 or 2 minutes, not more than that. It will be focused. So within that time, you have to identify, you have to write the identifying features, at least 2. things are very difficult. I have seen students suffering from that. For that you have to practice. Don't take histology lightly. If you take histology lightly over here, you will suffer in pathology. Okay, so that is all. Today I will stop over here. Okay, so in the next class, whatever we have discussed, after that we will continue. Right?